Hi, everyone. My name is Estrella Martinez. Now it's time for our next speaker of this morning. This conference is in charge of Dr. Pablo Meyer. Dr. Pablo Meyer is the manager of Biomedical Analytic and Modeling Group at the IBM Center of Computational Health. He received his PhD in genetics from Rockefeller University. He is interested in applying algorithms and models to biological healthcare data, and specifically who events determining higher order, phenomenal behavior disease from regulation of bacteria, mitochondrial fractional control of cell death, circadian behavior and flights, and prediction of human olfactory response in disease. Today, we are here to listen to his talk on front cell touch clinic predictive model in system biology. Thanks for your time, Dr. Pablo Meyer.
Okay, hello. It's a pleasure to be here virtually in Mexico. I couldn't be there, but I'm dressing a guayabera and sitting in a Equipal, which is, I guess, as close as I will get to Mexico. Maybe also, I could start playing a little guitar, so I'm really feeling Mexico. But it's a pleasure to be here. Thanks for the invitation to this CISBio MX4 virtually. It's always a pleasure to give talks in Mexico, again, even if they're virtual. So today, I want to talk about the work that has been going on for the past 10 years in, especially in the DREAM competition. So DREAM are, crowdsourcing competition that started through the reconstruction of gene networks, but really then expanded into several fields around systems biology, and also even beyond systems biology. For the past 10 years, we've organized more than 60 challenges. And today I will focus specifically on the challenges that are related to systems biology, and trying to get them into a nice narrative that explains how DREAM is constructed, and I think the big impact that DREAM has had in the community. So I will also specifically focus on challenges that I have organized and that I'm still working on. But we'll mention others that are important for the field of systems biology. And by the way, we publish most of the results, so these reflections that you will see in a brief format, together with Julio Saez Rodriguez, who's also going to be presenting at this conference. He's a very good friend and fellow director of DREAM Challenges, and we've been working hand on hand on this. So today, the title of my talk is From the Cell to the Clinic, Predictive Models in Systems Biology. And really, I want to first start with a reflection of the two types of models that we see in systems biology. The first one is the forward models. So these are models where we already know, in a sense, the biological elements that we are interested in. So for example, the proteins, we have definition of the proteins, the RNA, the genes, the metabolites, the drugs. And what we want to do is we want to really kind of model how these work together, right? And we can do this through different types of models. Most typical ones, ODEs. We can include things related to biochemical networks, stochastic models, rule-based modeling. So there's a plethora of models that actually take and use the knowledge that we already have about a system to try to understand how all the pieces of the system work together, and also work in a dynamic way. And then there are the other types of models, which are the reverse models. And reverse models are more about, well, I mean, we know some things about the system, but we want to know even more. We maybe want to discover new ways, new concepts, new features extracted from data. And in order to do that, we build these reverse models, which are basically, the typical prediction machine learning models, such as decision trees, support vector machines, lasso regularization, and all types of neural networks, of course, even including the typical and most loved linear models, right? And so really the difference between forward models and reverse models is that for reverse models you need a lot of data. You need a lot of data, and you can do reverse modeling even in systems where you don't know a lot, right? You can set up a cross-validation fold with the data and try to predict something. So now I think in Dream, one of the things that we do in Dream is precisely mostly what we do are competitions where we evaluate reverse models. Now, why is that? Well, it's just because forward models are difficult to evaluate, right? You could think about what is the best forward model. Well, the best forward model is probably the model that predicts the best something, right? But as you know, predicting something doesn't mean that that's the correct model, right? It's an apory, right? If you predict something, it doesn't mean that the model that you're doing, I mean, it at least has to do that, but is it the correct one? We don't know. So what is the correct model that defines and predicts a biological system? Well, you could think that a correct model could be the most detailed one. And some people doing biochemical networks think that if you model every step of a reaction, then that will be the most correct model. Now, the problem with those models is that yes, you're modeling every step, but the problem is you cannot measure the parameters for every step in the reaction. And not only that, but also these huge models with many parameters, become hard to interpret. So as a physicist in general, what we like is models that are simpler, right? Just following the famous von Neumann quote, if you give me 4 parameters, I can fit an elephant. So we like simple models. The problem, and it is possible to simplify models, right? It is possible to do an analysis of which are the parameters that are the most influence the prediction, and maybe then do a surrogate model and evaluate that. But as you see, just in this brief discussion, it is very difficult to evaluate what is the best model when you're doing forward model, right? Is it the simplest one? Is it the one that you can interpret the best? Is it the one also that makes the best prediction? I mean, it comes into the realm of things that we're seeing now in machine learning, which is what is the best explainable model, and the whole field of, if you want, explainability in a sense. Now that's why in Dream we've done what we've organized is in general competitions that have to do with reverse model, because it's easier to ask a problem, set up a problem, divide the data into a training set, and then use the test set for evaluating the participants. And then you do get into a part that sometimes can be murky, which is to decide which metrics to use to evaluate the models, and how many metrics, and also whether participants should know what these metrics are. So typically we try to be transparent, and we try to tell which are the best metrics, but sometimes even us don't know all the quirks and problems that we can have with when we organize a competition, and so sometimes we can change the metrics, things like that. Now, so most of the Dream challenges have to do with reverse model, right? So predicting a subpart of the data set. Now, one of the things that has happened over the years that has been really nice, and I'll try to show in this talk, is that sometimes results from reverse models actually feed back into forward models, meaning that you find a new feature in the data set. You find that, for example, I don't know, some type of k-mers in DNA are useful for predicting the strength of promoter, right? And so then what you do is you extract that feature, now use it in a forward model. And then the forward model also, helps you generate new ideas that then can lead to setting up new reverse modeling competitions. And so you get this kind of nice loop where these two types of model that seem pretty disparate actually feed into each other. And this is something that has happened, and I will try to talk about this. So over the years, nicely, Dream has been able to, in a sense, go from the smaller scale to larger scale. So we started by, how do you activate the cell using different types of drugs, and how these drugs affect, phosphorylase, and how the signaling is made to then affect the nucleus, maybe activate a transcription factor that binds to specific sequences. These actually, transcription factor is not alone. It's part of a whole network of genes and interactions with genes. But in particular, it's embedded in a subset of interactions with other genes, and also the transcription factor is part of that. We then, once you have defined the networks and the context in which a transcription factor is activated, you can start predicting what its dynamics effects are going to be. Finally, these dynamics effects have, an influence of where the cell that you're studying lands and how it's interacting with other types of cells, and also kind of where it comes from and how it landed, into the tissue that it's part of. And once you have all this information, right, in the context of the tissue and about, how a cell works with all the other cells around it, you can start thinking about, going to further modeling the disease, in a whole organism, for example, in humans. And so really the voyage I want to do with you today is to go through all these steps that we've done through several dream challenges that specifically deal with one of the steps that I was showing. So for the first part, so here is, how do you express a gene? How a transcription factor interacts with specific sequences to then express a gene with a certain strength. And the first challenge that we did, I'm going to not talk about a lot, but it was published in 2011 in Nature Biotech, and it's about transcription factors binding in vitro to specific sequences in arrays. And we found basically how to model this. But the other challenge that I want to describe is one where actually what we did is, the collaborators, Danny Sevi from Rand Segal's lab, is it took sequences of promoters, different promoters. These were promoters of yeast, actually, of ribosomal genes. Ribosomal genes are interesting because, ribosomes, the proteins in ribosomes have to be expressed in a stoichiometric way. And some of these genes sometimes are expressed by the two alleles, or sometimes are expressed in one allele. And so how do you actually encode the stoichiometry through the strength of the promoter? So what they did is took each of these ribosomal promoters, they fused them to YFP, they inserted in the same site in yeast that were reproducibly growing exponentially, with control promoters that were not under the expression, they were exactly on the same site. And so basically now they could measure over time the level of expression in a very reproducible way for each of these genes. So what they obtained is a list of 53 promoters that need to be predicted on a test set, and we gave them 90 promoters as for the training set. And one of the tricks that we did in the challenge was that we didn't give any of the synthetic genes as training set. And by what I mean synthetic genes, these were just basically the same promoters where we induce some mutations, either random mutations or mutations that specifically would affect transcription factor binding sites. And this will have some consequences in the results, as you see. But we felt that should be the way, because people should be able to extract from natural promoters the values of expression. So with this data, more than 18 teams participated and submitted their solutions. And what you see here is, in green are the values of the promoter predictions. The average values, if you want, just normalized back the actual value of the gold standard value, so basically the test set value. And you can basically you're just calculating a mean average error between the predictions and for all the participants for the predictions and to the gold standard. And so what you see here in green you have the natural promoters, and in open circles you have the synthetic promoters. And so what you see is that the natural, so you have the 53 promoters here that they had to predict. And you see that the natural promoters are better predicted than the synthetic promoters. Not only that, but you see that the synthetic promoter prediction varies with the type of mutation, right? So for example, if you add a mutation to the data box that affects a lot the change in transcription, the prediction was worse. And you know, if you add a random mutation, the prediction was better than when you were adding a data box. So really, kind of the type of synthetic mutation that you were adding was affecting the prediction of the participants. And by the way, the best performing solution was by Geoffrey Sewell. One of the best models was by Geoffrey Sewell, and he used just a support vector machine that was using k-mers to predict the expression of the promoters. The next thing that we did is we basically just plotted the distance between, again, just order by which promoter was the hardest to predict to the which promoters were the easier to predict. And if you separate these, basically just choosing a threshold between in red the ones that are difficult to predict, and in green the ones that are easy to predict, you can see that these two populations are clearly separated in terms of how good the prediction is, here measured by Pearson correlation, for all the teams that participate in the challenge. So every point is one team, and it's the average prediction of the team for, you know, the worst 23 promoters and the best 30 promoters. So what you can really clearly see is that the promoters that are easier to predict, so these ones in green, every team predicted them better than the promoters that are harder to. So there was really an inherent difficulty in predicting these promoters that's independent of the type of approach that was used by any of the participants. Finally, another interesting observation is if you calculate actually the change again of expression of the promoter, so how much change the mutation that you induce in the synthetic promoter induce in the expression of the promoter. Well, the most change that it induced, as you see here. So if you go into this side here, the prediction is the mutation induced a stronger change, the more difficult the prediction was. So really, it seems that you know, participants couldn't really. They had good scores, as you can see, as you saw in the previous slide. You know, the Pearson correlations can be pretty high here for you know, a lot of the promoters. For some, you know, they're much worse, but the predictions are pretty good. But really, you know, the effect of the mutation was just missed. So this is interesting, right? Because it's telling you that if you don't look at the biology, right? If you don't look at the fact that the promoters that you're predicting have binding sites and have things that are important for and that are known biologically, right? Data box is, you know, a very known element. So if you just ignore that and have a completely, if you want, blind approach, data center approach, then, you know, you're gonna miss you know, the hardest part. But if you use your priors and try to understand, right, that mutations and changes in specific sequences can give a big changes in the expression, then you will do a much better model, right? So that's the if you want, in a sense, conclusion of this challenge is, yes, you're doing reverse engineering problems. You know, you're doing a reverse modeling. But even when you're doing reverse modeling, you should, as you know, think of your priors, in a sense, think about the biology underlying it in order to have better predictions. And just as an example, I mean, one of the things that we like to do in DREAM is to have the, what we call the wisdom of the crowd solution, which is, aggregating the solution from the teams. And one effect that you see here very nicely, so here in red, you have just the score of all the teams ordered by, how good they were. So the higher score is the better. So here you have the best performer, and then the scores just go down to all the 21 teams that participated. But what's interesting is that if you actually aggregate teams following this order, right? So this value here, this score is actually the aggregation of the prediction from the two best performers, and the aggregation in this case is just, get the average prediction for each of the promoters between these two teams. And here's between the first three teams, etc. And so what you see here is, well, two things. One is the prediction is pretty robust, right? So here in this prediction, you have averaged the prediction of all the teams, right? Even this team that was really bad. And what you get here is actually a prediction that's better than the best performer, right? So if you know nothing about the problem, just get the prediction of the aggregated values for among the teams, and you will get a very good prediction. But what's even interesting is here, when you average the values of the promoters and the aggregate promoters for the first four teams, you get a solution that's much better than the best performing team. Right? So this is something that I will not repeat, but this is something that we see time and again for all the challenges. So the conclusions for this is that participants use mainly sequence information, but not really the underlying biology. The algorithms were mostly based on k-mers, and as I said, an SVM algorithm was the best one. And they gave too much weight to the wild type sequences and didn't look into mutations that were induced. Now, as I said, what's nice about DREAM is, we organized this challenge about 10 years ago. Yes, this is type of old news, and back then we had data for about 150 promoters, and we're really happy about it. But what's great about DREAM is that we follow the technology and we follow how things change. And precisely 10 years later, just now, we are organizing together with Cardebor and Rafi Abdul this predicting gene expression using millions of random promoters. So we've jumped from 100 or so promoters to millions of promoters, and we're still doing this in the same organism in yeast. But the trick for this jump, this 10 to the 4 jump in the amount of data that we're using, comes from the fact that it's very easy to generate libraries of promoters that are just random. So you just get you can synthesize chunks of DNA that have 80 base pairs. You just add them into very similar plasmid to what I showed you before. You transform yeast. This in yeast, you just separate, using just facts, you separate yeast by the type of, the level of expression of these random promoters, and then you just sequence, your different beans, and you obtain all the different sequences. So now you can have, a projection from the sequence, the random sequence that are generated to the actual level of expression of the gene, exactly what we did before. But what's interesting is that given that you're doing such, 10 million random promoters, just by chance, you can actually include into your sequences real natural promoters, right? And so, for example, this one, which is a pretty common promoter, every about 250 base pairs, you will have by chance, one of these. At least one of these sequences. And there are some that are, much rarer, but even for the rare ones, in this case, for every, 10,000 base pairs, you will have just randomly by chance, you will have one of these transcription factor binding sites appear in your data set. So what's really nice is that now, just by basically pushing the numbers, to 10 million sequences, you can have information for all the existing transcription factors in yeast, right? And so now what's nice is that with this amount of data, you can really get into, next generation models, which are, deep networks, And in this case, as you will see, using a transformer neural network, which is really the state of the art in natural language processing, you can use a transformer neural network to try to predict these millions of promoters. And you can see they do pretty well. So the Pearson sperma are really high, 0.93, compared to a more biochemical model that was already pretty good, it's 0.9. But really using this transformer network, you're getting an even better prediction. Now, as you can see, there are a lot of promoters that are not very well predicted, right? So there's still room for improvement here. So that's why we organized this BIM competition that's ongoing now. And here you actually have the values for the predictions from the teams that are participating. So this is a leaderboard, and you see here, this is the transformer model that was published actually this year in Nature, from the group from Aviv Regev, and Carl was also part of that group. But there's still a lot of space for improvement, right? This is the overall Pearson and Sperman that are really high. But now if you take subsets of promoters, depending on what types of promoters you have, you can see that the scores are lower. So there's definitely room for improvement. So this shows how really, being ready for continuing with the evolution of technology brings you to now real deep learning in single cell biology, right? And you're able to model a lot of these promoters. So this is part of the loops that I'm saying that I'm trying to show about DREAM, right? It's not only about, oh well, you find new features and now you can model, do forward models in a different way, but also the same type of challenges that you're doing, you can repeat them while technology is advancing. And just to close on this part of the talk, another related challenge that we organized together with the ENCODE projects, the ENCODE DREAM in vivo transcription factor binding site creation challenge. And really the idea here was, if you use ChIP-seq data, right, and you have all these peaks and some peaks are smaller, and you basically what you do is you build up a ground truth by binarizing the labels. For example, in this case, well, probably there is some transcription here, but not on this side. And what we did is then you give participants different features from the sequences. You give a DNA accessibility by DNA-seq data, gene expression data, gene annotation from gene code, and even in vitro DNA shape features. So you give all that, and you give data from 109 cell lines, 13 different types of cell lines, and 12 different transcription factors, typical and general transcription factors as you see here. Then you ask them to predict the binding sites for transcription factors in the same cell line and across cell line, which is, of course, much more difficult. The result of this challenge is actually pretty interesting because people use mainly K-mer models, included chromatin accessibility and transcription factor interactions and binding sites. But one of the essential elements for predicting, across cell lines was using the ATAC-seq data from, in order to have global chromatin, local chromatin accessibility. So this is the last, if you want, of the types of challenges that we have organized and are still organizing related to really how a transcription factor, both dynamically and statically, binds sequences of DNA and how that produces different levels of gene expression. So now that we know, in a sense, how an external influence can start transcription and which are the important elements, the question is, as I said, how this transcription factor functions in the context of a network of different genes. And of course, the first thing you have to do is to actually define and find what this network is, right? Once you have defined the network, you can now get into, how the transcription factor interacts with all the different parts and what are the consequences of these interactions. And so for that, we organized a few years ago, network inference DREAM challenge. As I told you, really DREAM started with these types of network inference challenges. And I think the one I presented here is really kind of like the top of the mountain from all the gene network challenges. I was not really involved in it, but this was mainly organized by Jim Costello and Gustavo Tolowitzki and other members of the team. But it's really kind of a culmination, right? So as you can see here, it's a 4-part challenge where you use in silico, and this is something that you will see that we have used sometimes, right? So when you don't have data, you kind of fabricate data in order to, that looks like biological data in order to really be able to evaluate your algorithms. So we use, different in silico data from E. coli, data from, once again, yeast, and a very special one, which was data from Staphylococcus aureus. Now, bacterial networks are much easier. So really what you do is, you have 296 transcription factors, and you have to say, how they interact with all the genes. So you have to build this gene interaction network. And for that, you use microarrays of values of gene expression in different number of conditions so that you can see the perturbations that happen. Right? And so you do this. And so now the idea is that, you can now insert inference methods, and 29 methods were applied by teams. All the teams were blinded. And we also tested 6 off-the-shelf methods in order to, build all these different networks. And as you see here by the colors, different approaches were used for doing this inference, from regression to mutual information correlation Bayesian networks and other types of, kind of in-house methods. And so what's interesting here is that, you can see by the type of method here which ones do better. And you also see for different types of data here. This is the overall where you average across all different types of data. So you can see here, as I told you, in silico data is of course easier than experimental data. E. coli data is easier than eukaryotic data. As you can see here, it was very hard to infer the networks in Cerevisiae. The other thing that we observe very often also in DREAM is that there's not always a single method that's better than all the others. Although here, kind of like in-house methods seem to be a little better than, regressions that seem to go second, or also mutual information that was pretty good. Now, one of the things, again, that you see here is that the community prediction, so where you aggregate all the solutions, and you can do that because, how we ask participants to send their predictions is by just ordering the edges by or by confidence, right? And so you can now average across participants, and basically it's a nice way for selecting aggregating the top edges for each of the predictions. And again, so what you see again is that if you aggregate all the methods, you have a pretty robust one. And here, if you start aggregating more and more methods in a kind of random way, you see that the more methods that you aggregate, the best results you get. Which again, this is the kind of wisdom of crowds, and this paper, Nature Methods, was really kind of the cornerstone of this wisdom of crowds approach. Now, what's interesting is that one of the networks I didn't talk about is this Staph aureus network, and I didn't talk about it because actually we didn't really have a gold standard for Staph aureus because it was kind of a much less studied than E. coli. But then what was interesting is that what we did is we precisely decided, right, with this background, that we were gonna obtain the wisdom of the crowds network, reconstruct network from Staph aureus, and we were gonna look at how all the different genes were clustered and how they were clustered by different types of function. And so when you do that, you see that very nicely you obtain wisdom of the crowds network of Staph aureus, where all different biological function cluster in different parts. And so for most of the genes, this was not known. So this is really telling you now information about the genes. So again, as I said, this kind of reverse challenge is generating knowledge that now can be used in a forward model. Right? This is one of the examples, clear examples of how this reverse modeling can generate biological knowledge. And as you see, in all the these I think 4 challenges that I've described until now, this is really the case. Okay, so now that we have defined transcription factors, have defined in which context these transcription factors work, can we actually predict now their dynamical effects, right? Can we predict what happens when you perturb over time transcription factor, the effect of probably one of these drugs, what happens, and can we actually model? So in order to do that, we really went back into this idea that we use in silico data. And so what it is, we built these networks that look like, a cascade, if you want, signaling. It's a network that has, in this case, 9 genes and 13 edges. It is determined by 45 parameters, and I'll go a little bit more into details of how we model that. And we use this Tinker cell. And so really what we did is we just did using, Michaelis-Menten type of equations to model how a gene is activated. So a gene can have an activator, a repressor, it has a promoter that has a certain strength, and it has a ribosome binding site that helps, the translation from once you have transcribed your gene, you translate it into a protein. And so really, so what we have here is just basically, an activator that you see. This is the known form of the equation for Michaelis-Menten, activator with has a Hill coefficient that we change. The repressor again has a similar form, but there's no the numerator is just a 1. The transcription is really just proportional to the promoter strength and the value of the activator and the repressor, and then the translation. So the protein level is going to be just dependent on the level, the amount of RNA, the amount of RNA just multiplied by the strength of the ribosomal binding site. So with this, we can really generate, a simulation of how a network such as this one will work, and we can also induce perturbations into this network. So for example, what happens if you change the value of the activator times 2, and here the repressor times 2, and then this other activator times 10. And this affects these 3 genes. And so what we asked is, what is the value, what will be the value of expression of these proteins if you change the values, if you somehow induce a perturbation. This is the dynamics that we found. So there's some interesting dynamics, right? Not everything is boring. Some things go up and down. Some things, plateau. Some of these different 9 RNAs for this first model. One of the tricks that we did is we actually wanted to be, if you want, as close as we can to experimental setup. And so in a sense, we gave credits, like when you go to the casino. And so they had 10,000 credits. And with those credits, they could buy experiments. So they could perform experiments where they would, delete a gene, erase an RNA, or change the strength of the binding of the promoter. And these were the values, the cost of the experiments, 800, 350, 450. And also, you can obtain experiments. So you could measure mass specs, you could have the value of the proteins, you could have an array in order to have the value of all the genes, or you could do, time courses of proteins. So each protein costs $400. So in a sense, you have a credit, you have a limited set of data, but you can choose the data that you want. And so what we did is we asked participants to give us the values of the parameters for 4 or 5 parameters that make up this network. And we calculated, just using a distance. And this was a hard problem, but you see that here the best performer actually had a score that was statistically significant. And then we also, as I said, ask to predict the values for the perturbations of the proteins. Once you have the network, you can now perturb the different promoters. And we calculated, right, for each time point, what is the distance between what they predicted for the value of the protein to the actual value. And in this case, this was a kind of an easier problem than thinking the parameters. And you see that the best performer had a score, a very, very significant score compared to all the others. By the way, we have about 12 teams that participated to this challenge. And this is the overall score, just calculated as, you know, the multiplication of the p-value for each of these two scores. And you have here the values for, you know, 12 participants in the challenge. Now, what's interesting about the protein perturbations is that when you look at, you know, how good the participants were, you see that in green, this is the actual value. And in blue, you see the solutions by all different participants. So some are very good and some were really bad. And again, what's really interesting is that even in a situation as this one, you see that if you want middle of the crowd solution, blue is much better, right? Interestingly, also the distance between the parameters and the actual values of the proteins are not really correlated. By the way, this is the best performer, Woru. Was an MIT now he has his own lab. And then we can also look at, you know, which parameters were the most important for the predictions, right? And again, this builds knowledge, right? And these are the parameters that were the most important. So clearly, you know, ribosomal strength was up there, and then you know, some of the KDs for the values of the expression in protein 13. Just very rapidly, we also could analyze, you know, which experiments were the most used, and in this case is you know, the deletions and some extra wild-type data were used a lot. We can actually look at how the best performer bottled these experiments and try to extract some, you know, knowledge out of this. Yeah, so it's a pretty complete challenge, as you can see. And there's a second part of the challenge I didn't talk about, but it's about trying to find extra edges on a network with some of these dynamic data, and that's a much harder problem. Okay, so now again, as I said, you know, dream challenges inspire us to do new ones. And this dynamic challenge inspired us to actually do a challenge together with Jonathan Carr, who's at Mount Sinai, to try to see if we could actually find parameters from a whole cell model. I'll be just very fast in this part because I want to reach the two last parts of my talk. So Jonathan Carr published in 2012 from Marcus Cavert's lab, when he was in Marcus Cavert's lab, the first model of these mycoplasma, which is a pretty simple bacteria in the sense that you know, there's not a lot of RNA regulation. So he did a 9-hour simulation of cell division, you know, 1 second resolution, and the model had 28 cellular processes, metabolism, transcription, translation, and it had 1,500 parameters. So what we, you know, and it predicts the copy of each molecule, the position of each DNA and RNA protein, the cell mass, and more. So what we asked is we modified 30 parameters in this whole cell model, which had to do with RNA polymerase promoter affinities, RNA half-lives, and metabolic reaction kinetics. We didn't say which parameters they were, and what was interesting is that it changed a lot the half-life of the cell division. So it had a very big effect. These were the modifications that we did. And so then what we asked is, you know, given the data, can you actually find which parameters were changed? And the best performing team is a whole cell modeler. Crux had an approach which was basically using the values of the parameters through some experiments. So, you know, don't look at Crux. But whole cell models were basically able to bring the values of the initial parameters, you know, an order of magnitude closer to the actual values of the parameters. And these are basically also, the same way as in the previous challenge, we scored based on the distance of the parameters and also the distance of the experiments. And you can see here how again the actual values for the data are better than the predictions for the parameters. And you can see here how parameters that were changed were harder to predict, and of course parameters that were not changed in green. And these are, all the different types of parameters that were modified. And this really, I think, brought a kind of an interesting approach where, Alex Williams, who was doing neuroscience at that time, just thought about, doing a differential evolution part of how to find these, to variate all the different parameters, and then using random forest to try to find which are the best parameters, and basically do a loop between these changing randomly the parameters, find the population, then find the parameters who could be the best values using random forest. Okay, so now that if you want to have a model of a whole cell, the next step would be to predict, where do the cells go. And for that, we organized a challenge that had to do with developmental biology. So, in developmental biology, we know that some genes determine where the tissues are formed and where they are. And in particular, in Drosophila melanogaster, is very interesting because, you have this segmentation of the embryo, and each of the genes kind of codes for a different segment. So what we did is we obtained transcriptomes from single cells, 295 single cells. And this is in collaboration with Nicolas Rogers lab and Julio Rodriguez with his postdoc, Kaneski. We organized this challenge, and also, Gustav Stolovisky. And so the idea is, can you place these cells in the embryo? So how do you know, anyway, where the cells go? Well, what we did is we used data from in situ fluorescence, and we determined that the place of a cell in the embryo will be the one where the highest correlation is to these 84 driver genes we are now looking for. And so what we asked participants, and can you actually use a small number of genes to try to predict where the cells go? We had a very nice participation using all types of different approaches, from random forest to neural networks, lasso, PCA, to select which genes were the most important. And once you did that, then the prediction is just based on a very similar metric as what we did, using Matthew correlation coefficient to find, once you selected the genes, you can find the highest correlated position to the actual values of the gene. And so what's interesting is that, so we did 3 sub-challenges, 60 genes, 40 genes, and 20 genes. They had to sub-select. And what's interesting is that you see that, some genes were, this is just 84 genes ordered by how many times they were selected by participants. And you see that, there's some coherence between challenges. And also these genes were not, clearly not selected randomly. What's nice is once you have these, for example, 60 selected genes, you can cluster them, and you see that they nicely cluster also, in their expression, but also into where they are expressed in the cell. And so here you can see the different clusters for all the different genes that you select. So it seems that each gene is kind of like sub-selecting a part of the embryo. Now, if we want to look a little bit more at their properties, so we calculate the entropy for each of these genes. And we also calculate the spatial autocorrelation. So spatial autocorrelation is just to see, where are these genes expressed? Are they just in one part of the embryo? Do they really don't have any correlation? Do they have a negative autocorrelation, just as checkers? This is just the formula for spatial autocorrelation. So what you see here is, for the entropy, in green, you have the entropy for all the 84 genes. In red, you have the entropy for the selected genes. And you see that the more genes you select, the higher the entropy. So you're selecting genes that have high entropy, so that are expressed, in the higher number of cells. And what's also interesting is that the spatial autocorrelation, it becomes more and more negative. So it is already more negative, but the more genes you select, you really are selecting genes that have negative value for autocorrelation, meaning that you're selecting genes that are expressed everywhere in the embryo. So you're really trying to maximize both the entropy and the spatial, and minimize the spatial autocorrelation. These are the interesting genes. I'll just skip that part. And so now what's interesting is that if you look at the subselected genes here in green, this is for when you have to select 60, 40, or 20 genes. You see that the genes are selected, at least the sub-challenges in green match to pairable genes in Drosophila, and the yellow ones match to gap genes. So it really seems that in this kind of blind approach, you're selecting genes that have biological importance, right? So in a sense, you're kind of recreating or finding again through this more data-driven challenge the genes that are biologically important, right? So it's a nice, if you want, way of rediscovering the wheel, but also seeing that your method is sound, and now being able to apply it to, if you want, other organisms. And indeed, the method works pretty well. You can, place cells through the genes in the correct segmentation. And this is just a kind of follow-up paper using similar approaches. Okay, so I have a few minutes left. This is, I think I can't go through all the details of this challenge, but as I said, the next step that we wanted is to build, how are these tissues built, right? Where do all these cells come from? We know how to place them, but where do they come from? What is can you rebuild these cell lineage trees? And cell lineage trees, in a sense, are similar to, evolutionary trees. And there are already existing methods for building evolutionary trees. The difference is that evolutionary trees have very long, if you want, genomes and very few organisms. In this case, with the cells, you have many, many cells, and you only have very small sequences to match them. And these sequences are already induced in a cassette and mutated using CRISPR, right? And so the idea is, how can you rebuild this tree? We did 3 challenges. One that used in vivo recordings, so you could see how the cells divide and then read in situ their arrays that encode different DNA sequences. And so the idea is that you could now, with this, having read the sequences, you could rebuild the tree and find the matching tree. The second challenge was actually using C. elegans, which is known to the cell division tree of C. elegans. The cell lineage has been characterized actually with a microscope. And so with that, we generated in silico values. This is how we scored the challenge using 2 different metrics. This was pretty original. Here is the best performing method. I won't go through the detail, and also the results have just been published. But basically, what's interesting is that we were able to, enhance the methods for reconstruction of cell lineages, something that had never been benchmarked in the field. So now, as promised, just the last part of my talk, and this will be a very, very brief one. It's just a single slide, but it entails something that's very dear to all of us, and this had to do with trying to predict hospitalization in pediatric colic. So what we did is my team participated this time. So this time we became participants to a challenge organized by NCATS and SAGE. So NCATS is a National COVID Cohort Collaborative Data Enclave. And the idea was they gave us the data from 2 million positive patients and 6 million patients in the NCATS data set. And from those, we had to predict hospitalization of pediatric patients. So there were 2,000 hospitalizations, 80,000 children were hospitalized because they were COVID-19 positive. And so using all their electronic health records, we were trying to develop a model to predict what were the important features for their hospitalization. And actually, we use a light GBM to develop a decision tree model. And, interestingly, geographical, so all these X4s are geographical position, demographical data of where these patients live, but the amount of lymphocytes. So the less lymphocytes you have, the better chances of not being hospitalized you have. This is just the SHAP values for the features. The age is important, and this pediatric Charlson index, which actually lists just a set of predetermined conditions that can lead to disease. So these are comorbidities. Body weight was important. So you can see that really, using a public data set and using one of these approaches for building models, you can really, obtain things that can have now impact, right? So I come to the end of my talk, and I think I've showed you how organizing challenges in citizen biology over the years not only helps evaluating in an objective way the submissions, but also being able to generate new knowledge across all different levels of this. So thank you, and if you have any questions, these are actually just a list of all the collaborators through the years.
Thank you, Dr. Meyer, for your fascinating talk, and we move to the question section.
Now it's time for the short talks. In this section, we will learn about the most outstanding works of the poster section by students and researchers.
Hello, I'm Pablo Meyer, and today I want to talk about my multidisciplinary project with titled Long COVID Treatment by Polymerized Type 1 Collagen Changes on the Physiological Network. First, I would like to present my collaboration network. In this graph, I represent all the people to work in this project, but the leader of this project is the Dr. Ana Leonor Rivera. Well, the human body is a complex system because collectivity pattern is emergent the organization of nonlinear between different organs. One form to study this network is using the correlation matrix. In previous work, had been reported that the correlation matrix is different between men and women. In this picture, I show you how look the physiological network. This is the woman network and the men network. We can see that the woman network is more connected than the men network. The physiological network is affected by disease, in this case, by COVID. In this example, in this article, propose or show how the correlation matrix is affected by COVID. Here is an example of the patient, the main patient, the correlation matrix, and this is the woman patient. We can see that the COVID affected loss in a lot of connectivity. One treatment by COVID is polymerized type 1 or PTAC. Exists a lot of articles that report that this treatment is affected different biological systems. However, we cannot not know how affected the physiological network. The objective in this work, we want to evaluate the effect of PTAC and placebo in the physiological network in patients with long COVID. Furthermore, to study sets difference in the correlation network and of the correlation matrix in post-treatment days. The datasets were provided by Instituto de Ciencias Médicas y Nutrición Salvador Zurita. We had 84 patients with COVID. These patients were selected randomly in the placebo group on PTAC. The blood samples were recorded in the baseline 1 day before to the treatment and post-treatment days in the day 1, 15, and 90. In order to study how changed the physiological network along to the post-treatment, in the data analysis only we use the patient we complete the post-treatment days. For network construction, we use the Spearman correlation to try to understand the interaction between different physiological variables. These correlation matrices were calculated in the baseline and post-treatment days. The first result is the physiological network for the placebo group. In the panel A, correspond to the older people or the patient, panel B, the main patient, and the panel C, the woman patient. The black line divided the baseline and the post-treatment days. We can see that in the placebo group, all the connections are very strong. This is the same for the three groups. In the case of the PTAC, it's the same order, all the subjects, men and women. And we can see that the first day post-treatment, the correlation increased, and then this correlation decreased. In order to quantificate the correlation between the network, between the baseline and the post-treatment, we calculate the correlation between the network in baseline and post-treatment. This is the result for placebo and PTAC treatment. We can see that the PTAC treatment decreases more faster than the placebo group. In conclusion, the physiological network of PTAC group increased the correlation in the first day post-treatment, and then decreased this correlation. In the placebo network, always maintain high correlation, and the correlation between physiological network in baseline and post-treatment decreases more in PTIC than placebo group. And the Goldman network is more affected in the case of the placebo group because maintain high correlation between the baseline and 90 days post-treatment that the network made. Thank you so much for your attention. Please let me know if you have any question or comment, and we will see in the poster section.
Hey, I am Russell Morales. I am currently a postdoctoral fellow at the Center for Research and Advanced Studies, or CINDESTAP, in the Monterrey unit. It's a pleasure to share some findings about our research. These findings were by Humberto Montesinos and the Dr. Jesus Rodriguez, that is a principal investigator of our lab. I'm going to start by commenting that the nervous system transmits electrical signals to the heart across neurons to regulate its function. Numerous researches have suggested that the nervous system and the cardiovascular system appear to leverage the noise or variability to improve information transfer. So noise added to stimuli may change the performance of the excitable cells, such as neurons or cardiomyocyte in heart. The role of noise or noise addition in the signal is explained by stochastic resonance. Stochastic resonance is a phenomenon that allows the system to improve its capability to detect signals when a limited amount of noise is added of signals. For example, in the cardiovascular system, some research have suggested that this phenomenon could enhance the homeostatic function of the human blood pressure. Recently, in our lab, demonstrated that the noise added on the signal enhanced contractile response coupling in isolated mouse heart and enhanced calcium dynamics in cardiac cells. However, few research have addressed in the fact that the noise can enhance cardiomyocyte shortening. Based on the above, we formulated the following question: how can affect noise stimulation on cardiomyocyte shortening? To respond to the question, first, ventricular cardiomyocyte of mice were isolated from a heart with retrograde perforated the Langerhans system. This is a representative image of an isolated cardiomyocyte. For electrical stimulation, pulse trains created from a function generator were used, transmitted by 2 platinum electrodes, where it is possible to control amplitude and the frequency, as well as the intensity of additive white noise to the signal by increasing the amplitude of the white noise by 10 to 30% of the amplitude with respect to the original signal. To obtain the temporal recordings of the contraction, we use the fast camera or ion optics. We evaluate the effect the noise added to signal in shortened response at 0.2, 1, 2, 3, 4, 5 hertz, and 10, 20, and 30% of noise was added to signal. We measured the shortening, such as percentage of shortening, velocity of shortening, and velocity of relaxing. In the control group of the blue lines, you can see that the parameters of shortening decrease as the frequency increased. However, when the noise was added to the signal, increased the response of the shortening in the third parameters only at 5 hertz. When a detailed analysis was performed at 5 hertz, we observed that with addition of 10 and 20% of noise in the signal, there was a significant increase in the shortening velocity and the percent of shortening and velocity of shortening and relaxing compared to the control group or without noise added. According to this finding, this project has improved our understanding of variability or noise role in the electrical stimulation in cardiomyocytes. The noise plays a key role in the cardiomyocyte shortening, and this data in the future associated with the clinical data and test new therapeutic alternatives, such as the condition of variability in the electrical stimulation. Thanks.
Hello, good morning, everyone. My name is Esbeiri Garcia Flores. I am PhD student and I'm going to present my project entitled DNA methylation levels of the TBX20 gene promoter in pediatric Mexican patients with congenital septal defects. This project was carried out of the Genomic Lab of Department of Molecular Biology of Instituto Nacional de Cardiología, directed by PhD Nancy Perez Hernandez. Congenital heart defects are defined as defects in the structure of the human heart during embryonic development. During cardiogenesis process, several transcription factors have a key role in the regulation of the heart formation. For instance, TBX20 gene, among others. Recent studies have related these transcription factors with different types of congenital heart defects. The gene regulation reported for these pathologies involved mainly mutations, polymorphism, and epigenetic. Regarding epigenetic, the main regulatory mechanism studied is the DNA methylation. However, up to date, research on DNA methylation of the TBX20 gene has been largely patients with congenital septal defects. For this reason, the aim of this research was to assess the association of the DNA methylation levels of the TBX20 gene with development congenital septal defects. We performed a pyrosequencing analysis to evaluate methylation levels of the TBX20 gene promoter. The sequence includes 7 CG sites in 48 patients with septal defects and 104 individuals with patent ductus arteriosus as comparison group. In addition, we performed an in silico analysis using bioinformatic tools to identify the potential binding sites for transcription factors in the evaluated sequence. And we developed a protein-protein interaction network for TBX20 protein. In this slide shows the results. In the figure 1, we presented the DNA methylation levels in the study groups. We found higher methylation levels in patient group statistically significant to compare with controls. In table 1, we identified the association the highest methylation levels with a significant increased risk of developing septal defects, with an odds ratio of 4.7. Regarding the in silico analysis, we identified binding sites for these transcription factors in the sequence. In the literature, these factors have been associated with congenital heart disease. On the other hand, in the protein-protein interaction network for TBX20 protein, we showed a network with a functional enrichment analysis, where TBX20 interact with another transcriptional factors involved in different stages of heart formation associated with congenital septal defects. In conclusion, these results suggest that high DNA methylation levels of the TBX20 gene promoter are associated with the risk of developing congenital septal defects. And bioinformatics tools is a good strategy to perform analysis by computer simulations. Finally, I would like to thank the support of different departments of Instituto de Cardiología and the patients of this project. Please, if you have some questions, you can contact me in the poster session. Thank you.
Hello, good morning. First of all, I'd like to thank for the invitation to this symposium. My name is Arturo Kensuke Nakamura Garcia. I'm a PhD student at UNAM, and I do my research work in the group of the Dr. Jesus Espinal at INMEGEN. The title of my work is The Network Structure of Hematopoietic Cancers. In our group, we employ network theory to study different types of cancers. And specifically, we use co-expression networks, which are made up of nodes, which represent genes, and the links between the nodes that represent co-expression interactions. This means that the pair of nodes connected have a similar expression pattern. And so here we use RNA sequencing samples from the Cancer Genome Atlas of different types of hematopoietic cancers. We have two groups of acute lymphocytic leukemia, one group of B-cell, another of T-cell leukemia, another group of acute myeloid leukemia, another of multiple myeloma, and another control group of normal bone marrow samples. And so all of these samples were processed in order to obtain an expression matrix. And then we employed an algorithm called ARACNI, which calculates mutual information values between the genes. And so these were the values of that we used to construct our networks. And so one of the main things that we wanted to know was the interchromosomal regulation in these types of cancers. In previous works of our group, we have reported the loss of interchromosomal regulation in several types of carcinomas, but we haven't analyzed these in hematopoietic malignancies. And so in this graph, you can see in the x-axis different cutoff points of the networks. We have the top 100 interactions, then the top 10,000 interactions, and so forth. And in the y-axis, we have the fraction of intrachromosomal interactions. This is the fraction of interactions between genes of the same chromosome. And so you can see that in the four phenotypes of cancers, the fraction of this is more elevated than in the control samples. And also you can see this phenomenon in the visualization of the network. Here I show you the network of multiple myeloma. You can see each color represents a different chromosome, and you can see how the genes are clustering together according with the chromosome that they located. And you can see that in the normal bone marrow network, all of the chromosomes of the different genes are interacting. And here I show you the same myeloma network, but here I show you the differential expression trend of each of the genes. I know this analysis we made this using DSEQ2. And another thing that we wanted to know was how the different RNA biotypes were interacting with the network. Because in the previous works, we have only used gene coding RNA. Here we used all of the RNA, or the different RNA biotypes that were available in the network, in the samples. One thing that really caught our attention was the presence of these black clusters of genes, which are genes of pseudogenes. And so we calculated the different types of links that we could have in the network. Here in these plots, you can see in the green bars the proportion of interactions between only pseudogenes. And so in the four types of cancers, there is an elevated fraction of these types of interactions. We also wanted to know what was the similarities and the differences between these four phenotypes of cancer. And so we took the four networks at the cutoff point of 100 interactions, and we intersected them in order to identify the interactions that were common in the four phenotypes. So we obtained this network. Here I show you that intersection network, but representing the different differential expression trend in each phenotype in comparison with the same control group. And so after doing our differential expression analysis, we used an algorithm called HiDF, which identifies communities in the networks. And those communities were introduced into Cluster Profiler to correlate the communities with different biological processes. And so here you can see all of the biological processes that are common for to the 4 phenotypes of hematopoietic cancers. And also you can see that there are similarities and differences in the differential expression trend. For example, these 3 biological processes are related with erythrocyte development, and the 3 of them are mostly downregulated. That could be somehow related with the anemia that these patients often suffer. But there are also several differences, for example, in cell division and in other processes of immune system. And so our main conclusions are that the loss of interchromosomal interactions is present in hematopoietic cancers, and that there are an elevated proportion of interactions between pseudogenes in these networks. And also that there are several topological similarities between these 4 types of hematopoietic cancers, but there must be differences in how they are regulated, and so there must be functional differences between these 4 phenotypes of cancers. Well, that will be all for the presentation. Thank you so much for your time. And if there are any questions, I'd be happy to answer them at the poster presentation. Thank you.
Hi, my name is Cristian Padron. The work I will show you is diffusion on PCA human manifold to denoise single-cell RNA-seq data. I'm a PhD student from the Human Systems Biology Lab, and the head of the lab is the PhD Osvaldo Reséndiz Antonio, here in the National Institute for Genomic Medicine in Mexico City. Basically, here I will show you a manifold learning method that we developed using single-cell RNA-seq data. Well, single-cell RNA-seq is a technology that permits to obtain the transcriptome profiles from individual cells. Unfortunately, this data is difficult to handle because first, it has a lot of missing values due to several noise sources. Well, that complicates analysis. And second, this kind of data is high dimensional. So finding insights is difficult because distances among cells in high dimensional spaces tend to be really homogeneous. So, and for that, manifold learning methods come to the rescue to deal with the high dimensionality of this kind of data, making a low dimensional representation such as this 3 UMAP projection that we all know that can be interpreted more easily. And this lower dimensional representation can be used for subsequent. For example, imputation methods such as MAGIC use diffusion on PCA to recover missing data. And diffusion by itself is a manifold learning approach. And however, there is always an inherent risk of imputing data based on PCA. But our approach, single-cell Phoenix, solves the major issues of using PCA initialization with PCA UMAP initialization. And we obtain a great improvement of using PCA UMAP initialization for diffusion and for imputation. For example, in this figure, we use bulk microarray data, and we corrupted by 80% of the values set to 0 values. We have evaluated the imputation performance by the correlation of the original data and the imputed corrupt data. So as we can see, in single-cell Phoenix, it's better than MAGIC in imputation performance. Also, single-cell Phoenix is robust against PCA dimensionality, something that MAGIC does not. And this is important because using more PCA dimensions, we can obtain a more variability that is involved in the system for a complete construction of a cell graph without the risk of introducing noise from a less variable principal components. So we went further and we visualized the denoised cell graph, aka exponentiated Markov matrix that has the information of cell similarities of Phoenix and MAGIC. And we use a multidimensional scaling approach for that. So in brief, this data set has several distinct cell phenotypes, the blue, the yellow, the green phenotypes. And for our surprise, we found that distinct cell types are really close to each other in the cell graph using PCA initialization with MAGIC. And this explains different users' comments about the oversmoothing or distortion of data with MAGIC. So imputing based on these default nearest cells will lead to inadequate results. However, using single-cell phenix that uses diffusion on PCA UMAP space, we observed that the local structure of distinct cell phenotypes is well captured. So having this kind of graph will lead to a better imputation. To see what kind of insights can bring imputation, well, we impute the breast cancer spheroid cell dataset. First, we project the data into these 3D PCA plots. As we can see, trajectories are more well captured with Phoenix than MAGIC. We found also that the highlighted genes of dense clusters present similar expression post-MAGIC, but with single-cell Phoenix reveals a more complex heterogeneity of cell phenotypes that MAGIC cannot offer. And also, using a gene set enrichment analysis of each cluster, and in this green heat maps, we can see that, well, MAGIC reveal or shows more or obtains several pathways in different clusters. But with MAGIC, we can see these pathways are over-smoothed. So as a conclusion, we can say that diffusion on PCA, aka MAGIC, is not enough to reveal local structure for imputation, but our method is well suited to capture local, global, and continuous structure for imputation. And finally, we have a version of microbiota data imputation based on a diffusion on a supervised UMAP space. You can find more information in our bioarchive using this QR code. So thank you so much for your attention, and I will gladly answer all your questions.
Do not forget to visit INMEGEN Facebook to watch the posters. You can make your questions in the comments box.